Hello, and welcome to this Integrated DNA Technologies webinar, Increasing Genome Editing Efficiency with Optimized CRISPR-Cas enzymes. My name is Sean McCall, and I will be serving as moderator for today's presentation. Today's presentation will be given by Dr. Christopher Vokulskis. Chris is a senior staff scientist in the Molecular Genetic Research Group at IDT. At IDT, Chris has managed contract research projects, led process development for CRISPR protein purification, and developed novel CRISPR proteins, including Alt-R S.p HiFi Cas9 Nuclease 3 NLS. Chris's presentation should last about 30 minutes, and following the presentation, he will answer as many questions as possible from attendees. The question and answer session will be conducted by Ashley Jacoby, staff scientist at IDT. As attendees, you have been muted, but we encourage you to ask questions or make comments at any time during or after the presentation by typing in the questions box located in the GoToWebinar control panel. Also, in case you need to leave early or want to revisit this webinar, we are recording the presentation and will make the link to the recording available on our website a few days after the presentation. We will also post the recorded presentation on our YouTube and Vimeo channels and post the slides on our SlideShare site. You will receive links to these in a follow-up email. So now let me hand it over to Chris for his presentation.
All right, thanks, Sean. Good afternoon, everybody. So I'm going to talk to you this afternoon about some work we've done to increase the activity of our entire suite of CRISPR-Cas enzymes. And I'll start by just giving you a basic overview of genome editing and some of the products we offer. And then I will move into the idea that the delivery mechanism of the CRISPR enzymes affects toxicity and off-target effects. I'll spend an awful lot of time talking about the Alt-R HiFi protein and how it can be used to reduce off-target effects. And then after each section, I'll try to go into some detail about how we've improved each one of these enzymes and show some real data. Finally, I'll talk about our Cas12a, or otherwise known as CPF1 protein, the basic workings of that system, and then end on the various improvements we've made to that system, and again, show some data. So just to get started, essentially what we're looking at are two different RNA-guided endonucleases that are used to edit genomes in living cells, and you're trying to introduce a double-stranded break as seen at the top here, and generally do one of two things: either facilitate homology-directed repair with some sort of DNA donor, totally replace the edited locus, or on the right-hand side here, allow the non-homologous end-joining pathway to form an indel, usually used for gene disruption. And so forth. And arguably, you could add a third branch on here for the newly developed base editors for both Cas9 and CPF1. And I'll start by just going over the basics of Cas9, again, an RNA-guided endonuclease. Here you're targeting a 23 base sequence, 20 nucleotides of which are encoded on the RNA. The PAM site for Cas9 is NGG. So anywhere on the genome where there's a G dinucleotide repeat, theoretically you could have a targetable Cas9 site. Cas9 exists in bacteria as part of a primitive immune system where you have a targeting CRISPR RNA or crRNA and a universal tracer RNA that hybridize together to form the functional guide RNA. Those have also been appended together covalently to form a single guide, or sgRNA. And this cartoon at the bottom depicts a functional RNP or ribonucleoprotein complex, where you can see that Cas9 in that sort of light blue color with the tracer and crRNA hybridized together, bound to the target site. And as I mentioned, you can use this as either part of the native two-part system or as the artificial single guide RNA. In IDT, we actually favor the two-part system for a variety of reasons. Here you've got two very short RNAs, which are not ideal to express as part of a plasmid delivery vector. They're really just too short to effectively terminate. And for the same reason, they're not ideal for in vitro transcription as well. However, we are an oligosynthesis company, so we can make them very easily and inexpensively by chemical synthesis. So for the CRISPR RNA or cRNA, you're talking about a 36-mer that's comprised of 20 bases of targeting sequence with 16 bases of constant region to anneal to the tracer. The tracer RNA is a bit longer, it's a 67-mer, but it's still very efficient for chemical synthesis. Because we're making them chemically one base at a time, we can actually incorporate modified bases quite easily. So it's easy and cost-effective to modify. That allows us to introduce modifications that help escape nuclease degradation as well as the immune response. And at the bottom of this section, you can just kind of see the cRNA on top annealed to the tracer. And alternatively, with the single guide RNA on the right-hand side, these are ideal for DNA expression cassettes because they're a bit longer. This is approximately 100-mer, so it's easier to terminate these as part of plasmid-based delivery systems. For the same reason, they're ideal for in vitro transcription. You can transcribe them in your lab and introduce these into cells. I would say they're relatively low cost to produce that way. Chemically, because of their length, they're relatively inefficient to make, although we can make them. Hence the higher cost. They are costly to modify, again, because of the length. And if you're going to produce them with in vitro transcription, it's very, very difficult or impossible to modify depending on where you're trying to introduce the modification. And we've also found that sgRNAs produced through in vitro transcription can also cause an awful lot of toxicity, which I'll show in the next slide. But I point out before I move on that the structure of these two things are very, very similar, and all that's missing with the two-part system are the covalent links. So just taking a really broad overview of what we found happens when using in vitro transcription produced sgRNA. We're just simply looking at the cells under a microscope and looking at morphology and abundance. And if we look at the left-hand side here, this is simply a microscope view of confluent HEC293 cells. They look off very healthy, and these cells are constitutively producing Cas9. In the middle, we've transfected 30 nanomolar of an IVT-produced sgRNA, and you can immediately see the lack of cell abundance and lack of proliferation, indicating there's some toxicity with this experiment. Now, if you look at the right side panel, here we've taken the same targeted guide, but instead of an sgRNA, we're using a chemically synthesized two-part system. And you can see these are largely indistinguishable from the cells only. And I'd point out that despite the differences in toxicity, both of these experiments produced equivalent levels of gene editing. So they both work, it's just we see evidence of toxicity for the IVT-produced sgRNA. And looking a little more closely at the transcriptome, on the left-hand side here, what we're looking at is a qPCR assay for a human normalizer gene, SFRS9. This is something where a control where we don't expect the levels to vary no matter what we do. And you can see all of the data points are on top of one another, the IVTs, the two-part, and the cells only, exactly as we would predict. But if we look on the right-hand side, we're looking at a marker for stimulation of the immune system, IFIT1. And you can see that the two-part system and cells only are indistinguishable, suggesting there's no induction of the immune system. However, there's a massive CQ shift to the left for the IVTs, indicating that the immune system is in fact stimulated when you use these RNAs. And IFIT1 is just one example of several markers for the immune system that we've looked at that show high induction with IVT-produced sgRNAs, and we just don't see this with the two-part system. Nevertheless, there are a lot of different ways to implement CRISPR-Cas9 genome editing in your laboratories. On the left-hand side, looking at the Cas9 protein, starting from top to bottom, you can purify Cas9 in your own lab. You can purchase it commercially. That can be delivered directly. You can either make or acquire a Cas9 stable cell line where the Cas9 protein is expressed at a constitutive level all the time. You can purchase or synthesize in your laboratory a Cas9 mRNA. This can be transfected directly with the right protocols. And probably the most popular is using either plasmid or viral vector-based DNA delivery system at the bottom. And it's essentially the same story for the RNAs. If you look at the right in green, From top to bottom, again, IDT, we favor the two-part system made by chemical synthesis. You can also produce these by in vitro transcription to make an sgRNA. That does work. You can supply a DNA fragment, be it a PCR product or a G-blocks gene fragment that encodes the RNAs that can be transfected and produce a successful genome editing experiment. And lastly, much like for Cas9 protein, you can supply the RNAs off a plasmid or viral vector. These can be mixed and matched however you want, really, and you'll get genome editing to work on some level. At IDT, however, we tend to favor ribonucleoprotein delivery, and that is taking purified Cas9 and chemically synthesized two-part, putting them together in a test tube to form the RNP complex, and then delivering that directly into cells. And part of the reason for that is simply just because of how easy it is. And it's a three-step process for Cas9, where at the top you take the universal tracer RNA and the specific CRISPR or crRNA, add them together in a test tube, heat and slow cool to form the guide RNA complex. That is added directly to purified Cas9 protein at a 1-to-1 ratio. The RNP is formed after about 10 minutes at room temperature, and then that can be delivered directly into cells by whatever your favorite delivery mechanism is. And we have protocols online for liposuction-based delivery. We have protocols for electroporation with either the Amaxa effector or the Neon system. There are also some user protocols for microinjection and other more niche systems. Again, all of these can be accessed from the IDT website. So it's very easy to use, but probably a more important reason to use RNP is because of the propensity for Cas9 to cleave off target. And I think that there is an awful lot of confusion in the field as to how much a person should worry about off targets. And a lot of that, I think, is going to depend on how you've set the system up, what guides you've chosen, and then also just how, what type of work you're doing and how much off-target editing events are going to matter. And I think whatever you're doing, there are some very simple ways to mitigate the risk of off-target events, starting with using RNP. And what you're seeing on the screen is an example from the GuideSeq approach. This is an approach that's widely been employed to determine the total repertoire of edited sites for any given CRISPR experiment. In this case, the authors are using an EMX1 targeting guide. This is a very popular site in the literature. And they've got all these different sites listed. So this is an NGS coupled approach. So the number of GuideSeq reads are NGS reads that correspond to the number of indels. So at the top with this filled square is the number of reads associated with the on-target site, and thankfully it's edited the most frequently. And then everything at the bottom, moving down from top to bottom, are all the off-target sites and the frequencies with which they are edited. What I think is kind of scary about this is if you look at the top two sites beneath the on-target, these are edited at near equal frequency to that of the on-target site. And for certain applications, that can be kind of scary. And I think one of the things to keep in mind with this experiment is that the authors were using plasmid-based delivery of both Cas9 as well as the RNAs. And we have found that to be a condition where you get the most off-target editing. And I can give you a lot of our own internal data, but I think it lends a lot of trust if I can show you data from other companies and then show that it falls in line with the data that we've produced. So our friends at Thermo have shown this nice experiment where they vary the delivery mechanism of Cas9 protein, and they essentially show that the longer it sticks around in the cell, the more off-target editing you get. So Cas9 protein by Western blot on the left, and the ratio of off to on target on the right. These are two known off-target sites for the guide that was used. So going from top to bottom, with a DNA or plasmid-based system, they find that Cas9, it takes a while for it to reach a steady state level in cells, about a full day, and then it doesn't seem to reduce it all the way out to 72 hours. And not surprisingly, in black, you can see that the DNA-based delivery mechanism is synonymous with the most off-target editing seen for both of these sites. If you look at mRNA, that seems to show that Cas9 protein appears a lot sooner, about 4 hours, and the levels do not dissipate that much, even all the way out to 72 hours, although arguably there's maybe a twofold decrease. And that's sort of an in-between level of off-target editing between DNA and protein here in blue. Protein, however, so if you're transfecting Cas9 protein directly, all of it seems to show up immediately after 4 hours, and then it just dilutes or degrades by growth beyond that point. And by 72 hours, it's virtually non-existent. And not surprisingly, in this red or burgundy color, RNP-based delivery is consistent with the lowest amount of off-target editing seen. So, we talk about it in the lab as sort of a fast-on, fast-off approach, where the longer Cas9 is around to cause mischief, the more off-target events you get. So yeah, that's the same sort of experiment we show in-house. And what we've done here is to compare Cas9 that is expressed in a constitutive stable cell line, HEC293 cells that always make it, to that of ribonucleoprotein delivery. And the readout for this in dark blue, this is the on-target site for this guide, so we're getting very good editing. And then these in light blue and gray are 2 known off-target sites for this guide. And you can immediately see that on-target editing is very similar between the highest doses of RNP and for the stable cell line, yet there is a lot more off-target editing seen for the stable cell line. And you can actually dilute out the RNP quite a bit here without sacrificing much on-target editing efficiency, and yet the off-target events go down even further. And it is only out to this half micromolar dose before you start to see a loss in on-target editing efficiency. So our finding is that simply switching your delivery mechanism to RNP can mitigate a lot of the risk from off-target editing. But we've also found that there are a lot of cases where, despite the fact that you switch to RNP delivery, there are certain persistent off-target sites that show up no matter what you do. And also, if you're somebody that's working in the therapeutic realm, even minute quantities of off-target editing could be a real problem. So what do? And one of the approaches to deal with that has been to try to predict off-target editing based on the guide sequence you've chosen. And these are 2 examples of the more popular prediction algorithms, the MIT algorithm and the Heidelberg CC-top. And what we found is that it is just an extremely difficult thing to accurately predict Cas9 cleavage sites. And for that reason, we do not think that this approach is quite ready for prime time. And here's some data to demonstrate what I mean by that. So this particular androgen receptor guide sequence that I'm showing at the top here, there's nothing about this sequence that seems repetitive or like it would be a huge problem. But nevertheless, if we look at the total repertoire of edited sites for a given genome editing experiment, we see an awful lot of off-target cleavage with this guide. So on the far left in red, that's the frequency of editing seen with the on-target site, and everything in blue and green is an off-target site. So there's quite a bit of off-target editing there. And I've coded in green the sites that are predicted by any SVM prediction algorithm. And what's scary is that you find that there are only 6 sites here in total that are predicted, representing less than 10% of the total off-target editing. And I'd also point out that the top most frequently edited off-target sites are not predicted by these algorithms at all. And these are probably the ones you care the most about. And I don't think that this is to say that all the algorithms are bad or poorly constructed. I think it's more evidence that it's an extremely difficult thing to do to predict off-target editing, and it's just, we're not there yet. I think one day we will be. But in the meantime, what more can you do to minimize the risk of off-target editing? Well, first of all, changing delivery mechanism is important. Second, there have been some other things that have come up in the literature to solve this problem, one of which is reducing the length of the crRNA from 20 mer to 19 or 18. We've tested this internally, and we found that in certain cases it can indeed help, but oftentimes you'll find an unpredictable reduction in on-target editing at the same time as a reduction in off-target editing. That's something you don't want. And it's been basically the same story for chemical modifications. There have been some papers that have come out saying if you put specific altered bases at specific positions at cRNA, you can minimize off-target editing, and we found it's a mixed bag. Finally, one of the things that has come out is the notion of mutant Cas9 proteins that are so-called high-fidelity Cas9s. And these are two of what I think are approximately 5 mutants present in the literature right now. The ESP-Cas9 1.1 protein from Feng Zhang's lab and the SP-Cas9HF1 from Keith Young's lab. These were fantastic studies done where they looked at the crystal structure of Cas9 and tried to rationally engineer alanine substitutions that would reduce the affinity for off-target sites and hopefully not for the on-target site. And we had set out at IDT to test these mutants because we knew off-target editing was a problem, make them in the context of our Cas9 and test for off-target cleavage as well as on-target cleavage with RNP delivery with the intent of finding the best and hopefully licensing and commercializing it. And we started by testing some of the guides that are frequently looked at in the literature, the EMX1, Hexsite4, and VEGF-A3 guides, and we looked at both on and off-target cleavage for both. These represent just a single off-target site for both. And we found that for EMX1, all enzymes were comparable to wild type for on-target, and all of them reduced off-target to undetectable levels, which is very good. But when we moved on to the Hexsite4 and VEGF-A3 loci, we started to see real problems with on-target editing, despite the fact that they were both good at reducing off-target. So this is quite worrisome, and we thought, okay, well, maybe we better take a broader survey of sites and find out, how big of a problem is this. And in fact, so we looked at several different sites, the HPRT, CTLA4, and PDCD1 loci, and we found that these enzymes really struggle to edit on target when using the RNP delivery platform. And I would point out that these enzymes were evolved under conditions where they have plasmid-based expression of both Cas9 as well as the guide RNA, so they were never made to work for RNP delivery. So I don't think it's a failing of these mutants. I just think we've put them in a situation where they were not designed to work that way. I think with plasmid delivery they probably work quite well, but for RNP we just needed a different solution. So these mutants were selected based on plasmid results, where you have continued and long-lasting Cas9 synthesis. It's always being re-expressed. We know that plasmid delivery is prone to toxicity and problems with the immune system, so we don't favor it. We like RNP. And we found that no existing HiFi mutant works well as RNP, and that includes more than just ESP-Cas9 and SP-Cas9HF1. We've also looked at HiFi Cas9, we looked at Evo-Cas9, we looked at X-Cas9, and we always find the same thing, a reduction in off-target editing at the expense of on-target. So we developed in-house a proprietary bacterial system to find our own HiFi mutations. And what we did differently is that we dually selected for reduction in off-target editing as well as the maintenance of on-target editing efficiency. So selecting for both things at the same time. And this is a basic cartoon demonstrating that. And I won't go into all the details here, but essentially what you've got is a high copy plasmid that expresses a bacterial toxin. So if this plasmid survives, the bacterial cells are killed. And on this plasmid is an on-target site for the guide we're using. So we're asking CRISPR-Cas9 to cleave every one of these plasmids so that the toxin is not produced. And simultaneously, we have a second plasmid, an off-target site containing plasmid, where you need this plasmid present to survive, since it expresses an antibiotic marker, a resistance marker, but it also has this off-target site. So Cas9 has to cleave all of these and avoid cleaving the majority of these off-target site plasmids in order to survive. And it's important to note that the wild-type Cas9 does not pass this screen. It would not facilitate any survival. So we made a random mutagenesis library of Cas9, passed it through this screen, pooled all the mutants, changed the guide and target sites, did the same thing, and ultimately we settled on a handful of mutations, evaluated them very carefully, and settled on one very specific combination of mutations that resulted in what we're now calling the Alt-R S.p HiFi Cas9 protein. And here's some data in two very, very different systems to show you the on-target performance for this HiFi Cas9 protein. And on top, we've got the two-part RNA system delivered by RNP lipoaffection, testing 12 different guides that target the HPRT locus. These are in standard HEC293 tissue culture cells. And we're comparing in dark blue the wild-type Cas9 to our HiFi Cas9 in orange, and then the two other literature high-fidelity proteins are in that light blue and gray color. And you can see at the vast majority of sites, the orange HiFi works nearly as well or as well as the wild-type protein, where the other literature proteins really struggle. And if we look at the bottom here, this is an experiment done by our collaborators at Stanford, the Porteous lab, and they are using chemically modified sgRNAs. They're delivering these RNPs into cells via electroporation, and they're looking at human primary cells and targeting 4 different clinically relevant loci. And then, whereas we were detecting with T71 cleavage at this top experiment, here we're looking at NGS. So very, very different system, demonstrating essentially the same result. The wild type and IDT HiFi work very, very similar at most of these sites, where the other literature HiFis fail. So these things, this HiFi protein works great on target, but it's probably good to show some off-target data. So these are 3 sites tested in literature, and we first looked at single off-target sites to see what happened. And EMX1, all the on-target is pretty good for even the literature mutants. All of the literature mutants and the Alt-R HiFi reduced off-target editing to undetectable levels, very good. Looking at HEC site 4, where the literature HiFis showed trouble on target, the Alt-R HiFi maintains. And importantly, the off-target editing is reduced where all proteins down to an undetectable level. And it's the same story for the VEGF-A3 locus. On target is maintained, all 3 HiFis reduce off target to an undetectable level. And we had thought that this is really good, but we really need to look at global off-target editing and really challenge the system and get a feel for how good is this protein. So to do so, we utilized a 2-step approach. And the first step was to use the guide-seq procedure to identify what the off-target sites were to start with, and then design amplicons around those off-target sites and use multiplexed amplicon-based NGS to actually do the sequencing at every one of those sites. So first using guide-seq to find out what the sites are, and then multiplexed NGS to sequence at every one of those sites. And we tested several different guides and looked at the results. And before I get into that, I'll show you just what standard guide-seq demonstrated comparing wild-type to HiFi delivered as RNP. And with wild-type, if you look on top, the first 2 VEGF-A3 and EMX1 experiments, there's only around 35% on target for both of these guys. They have a lot of off-target editing. The IDT HiFi at the bottom reduces that so that you're up to 80 to 81% on target. Moving to the right, there are 2 guides, DRHPR and the HPRT 38285 site. These guides, for whatever reason, are inherently low off target, so probably no HiFi needed. But the following 3 guides are sort of an intermediate level where you've got 79, 64, and 55% on target. And in every case, the IDT HiFi brings that up quite a bit. And through a lot of investigation with the guide-seq procedure, we come to appreciate that it is very, very good at identifying what an off-target site is. It's very good at giving you the total repertoire of edited sites. Where it begins to fail, however, is how quantitative it is when assigning an indel frequency to each locus. That's why we went on to using this 2-step procedure. So we took what we knew about these sites and we did the amplicon-based NGS, and we also upped the ante a little bit. We did both RNP delivery of wild type and HiFi, but we also created stable cell lines where we constitutively express wild type Cas9 or the HiFi mutant. These are essentially the same cell line, except we introduced the HiFi mutations into this wild type background. And in the log-based graph here, you're showing in this orange color the on-target editing. The far left is the on-target site. You can see that's quite high for this guide. And then all the blue sites are evidence of off-target editing at different frequencies, all rank ordered. So you can see for stable cell line delivery of wild-type Cas9, only 43% of the total editing occurs on target. And if we simply introduce the HiFi mutations, again, this is a system where we didn't develop the HiFi Cas9 for this. We developed it to work with RNP, but simply introducing those mutations brings 43% on target up to 97%. So quite an improvement there. And then looking at RNP delivery, this is an example where using the wild-type Cas9 by RNP, there are a handful of persistent off-target sites that you can't seem to eliminate. So you've got still 73% of the editing is on target, and then delivering the HiFi Cas9 by RNP brings that up to 99%. Again, this is one guide, this is the EMX1 guide, but if we extrapolate that to 4 different guides, you can see it's essentially the same story. A lot of off-target editing when we use wild-type Cas9 with stable cell line delivery. You can clean up a good deal of that in stable cell line context by introducing the HiFi mutations. Alternatively, if you use RNP delivery, you can use the wild-type Cas9. You still get some persistent off-target edited sites with certain guides, but you do clean up a lot of it. And then RNP delivery of the HiFi Cas9 gets you the rest of the way there. And in every case, we saw 99% or greater on target. So this is really a real example of the utility of this protein. And I think what we've done here is we've engineered something that works so well on target that even if you're a little uncertain of how much you need to worry about off-target editing, you can mitigate any risk by using the HiFi Cas9 delivered as RNP. And we've received some pretty great questions when presenting it at conferences or posters at conferences. And one that came up is you guys have introduced mutations into Cas9, but how do you know that you haven't changed the nature of the cleaved products in a way where using this mutant is going to affect the repair profile? So we actually took a look at that. And what you're seeing here is one example of a lot of different guides that we've analyzed, looking at the indel profile at the cut site, comparing wild type to HiFi. So in green here is cells only, and because there's no edited sites, it's going to be 100% at 0. There are a couple insertions that are present, and you can see that for wild type and HiFi, the frequency is basically the same. And the same is true for all the deletions that occur at this site. And taking it in the aggregate, looking at a lot of different sites, we can conclusively say that the HiFi mutant gives basically the same repair profile as wild type, and you can use it without fear that we're going to alter your repair profile. Very good question, though, and I think it speaks to the level of friendliness we have with the academic environment and how we sort of have a symbiotic relationship. Another question that has come up from that is, how well is the HiFi protein going to work for HDR experiments? And this is another set of experiments from our collaborators at Stanford, the Porteous lab, and they're trying to do correction of mutations at the beta globin locus. And what they're showing here is they have successful HDR with the wild-type Cas9 protein, but they're also showing that they're getting simultaneous indel formation at this off-target site. And they used our HiFi protein, and they're showing, again, good HDR, but significant reduction in off-target indel formation. But what they noticed is a modest reduction in on-target editing efficiency with our protein. And there are going to be certain sites where the HiFi Cas9 has on-target issues. It's not a perfect system. But what they were able to do is to add a little bit more protein and get the editing levels back up to that of wild type and beyond without sacrificing the reduction in off-target editing. So even in these cases where you do see modestly reduced on-target editing, you can simply add more protein and solve the problem without fear that you're going to introduce more off-target editing events. And this is something that we're really excited about because there are some big labs, both the Porteous lab, Jacob Korn, and a few others that intend to take this protein actually into clinical trials. So something we're very proud and excited of. And we're always asking new questions and trying new things and working to improve things. So I'm actually very excited to give you some data showing you the improvements we've made to these proteins. And we'll start with the wild type Cas9, and where we've actually increased the editing efficiency by making modifications to the linkers and NLS sequences that surround Cas9 delivered into cells. And this is an experiment where we've done gene editing to 12 different loci within HPRT, comparing our Gen 1 commercial Cas9 product in blue to the version 3 product in orange. And you can see at every one of these sites, we've increased editing efficiencies dramatically. This is a minimal dose. This is a 400 nanomolar dose of the RNP complex. And we've also put this series of changes into the context of our HiFi protein. And what you're looking at in this experiment is both on-target editing in blue and then all the off-target sites in various colors. So we've compared our Gen 1 and Gen 3 wild type and HiFi products in this experiment for on and off target. We've also looked at various suppliers, other suppliers of wild type Cas9 to get an idea of where our products performed in the field. And what you can see going from left to right is that our V3 wild type improves on-target editing at this site that was already pretty good. Since we've improved overall editing efficiencies, it's not that surprising that off-target cleavage goes up a little bit. We see that our generation 1 HiFi had a little bit of a non-target problem at this site, but moving on to our V3 protein brings the on-target editing levels even above where our wild type V1 product was. And importantly, we've still maintained a strong reduction in off-target editing. So we've got something now that works better than our previous wild-type product, but still is very low off-target. And if we compare that to all these different suppliers of wild-type Cas9, you can see that there's a lot of variability. There's some that work sort of average, some that work very poorly, and then a couple that work pretty well. And what I would point out is that our V3 HiFi product is actually equal or better than both of these options while having much less off-target editing efficiency events. So just to summarize this section, you can use RNP delivery as a first measure to mitigate off-target risk and avoid immune system stimulation and toxicity that's experienced with plasmid-based delivery methods. You can use it to mitigate off-target risk, but there's always the risk there are going to be persistent sites. And we're recommending that you use the Alt-R HiFi protein to sort of clean up the rest of these off-target cleavage events. And then finally, we've got this improved context for the Cas9 protein that we've introduced into every one of our Cas9 products. And that begins obviously with the wild type product. We previously offered 100 and 500 microgram quantities. We're now selling a larger 0.5 mg quantity. We also have this in the context of our HiFi. Again, we've introduced now a 0.5 mg quantity. We did not previously sell a dead Cas9 protein, but now we are going to have this available. And one thing I think is unique about this is we've quality controlled this not only to be dead for nuclease activity, but we also confirm that it retains RNA-guided DNA binding activity to make sure that you get an active and dead protein. And finally, we have our D10A and H840A Nickase proteins. We've upgraded those as well to put them in the V3 context. These are to produce targeted cleavage of just a single strand. And I direct you to our previous IDT webinar on the use of these products. And these will be available by the end of the month, actually. So these will be a replacement on our website for the V1 products. So I want to spend the remainder of the webinar giving a very brief summary of the Cas12a or CPF1 system, and then just quickly demonstrating the improvements we've made in this protein as well. This is another RNA-guided DNA endonuclease. The one that we are selling is from Acetaminococcal species. Unlike Cas9, which exists natively as a two-part system, this is a single guide RNA facilitated system where you can use a 41 to 40-former. The double-stranded breaks that are made with this system leave staggered ends instead of blunt cuts with Cas9. And the PAM site is located on the 5' end of the target instead of the 3' with Cas9. And the PAM sequence is actually a triple TV, or any triple T that is not followed by another T. So very, very different from the NGG PAM. And we've done an awful lot of work. Some of this is present in a previous webinar demonstrating length optimization of the guide, as well as certain chemical modifications you can introduce. And ultimately what we're selling as a commercial product is a 21 base targeted site. And you can also use RNP delivery with Cas12a or CPF1. And it's basically the same story as Cas9, where DNA or plasmid-based and mRNA delivery methods have higher off-target events than protein delivered directly. So we recommend RNP delivery. It's been reported that Cas12a is intrinsically lower off-target than Cas9. We're a little uncertain of that. We wonder if Cas12a is just a slightly lower activity nuclease than Cas9, and that way it appears to be that way, but the jury's still out on that one. And because this is not a two-part system, its use as an RNP is even simpler, where you really only have two steps. You can take purified or commercially acquired CPF1, add it to your guide RNA, form the RNP complex, and then deliver that directly. It's very, very simple and easy. We have not had consistent results with liposuction delivery of Cas12a, so we do not recommend using liposuction. However, we do have great protocols for electroporation for this system. And I think it's important to note that when doing your electroporation, we find that this system is actually dependent on an enhancer molecule that we sell. And these are the results demonstrating that fact. In blue, you have gene editing experiments with Cas12a where we don't add enhancer. And then in orange and gray, we have experiments where we do add enhancer. So we highly recommend and require using the enhancer with this system to get good results. And I mentioned that this is a triple TV PAM site, not the previously reported triple TN, and I think that these results nicely demonstrate that fact, where we looked at editing efficiencies at a lot of different sites in this green color. These are quadruple T PAM sites, and you can see those are not, the vast majority of those are no editing detected at all. Whereas for the other putative PAMs, triple TA, triple TC, and triple TG, those appear to be much better. So for that reason, we don't think people should select quadruple T PAMs for Cas12a. This is a summary of that data. So in orange, this is a ski slope plot showing the editing efficiencies of a lot of different Cas9 sites that we've tested. So this system works well. If we look at Cas12a, where we select any triple TN site, you can see that we end up throwing out a lot of those sites because they don't work at all. And if we simply subtract out the quadruple T PAMs and only select the others, you can see that that vastly improves the system here in gray, approaching the same type of efficiency you get with Cas9. And we're hoping to make that even better as we are testing greater than 1,000 sites for the production of an algorithm to better predict good cleavage sites for Cas12a. So lastly, I want to show you just very briefly the type of performance improvements we've made with this system, again, by introducing changes into the Cas12a protein, optimizing linkers, changing NLSs, and moving things around. Ultimately, what we've settled on is this V3 Cas12a protein, and the performance improvements seen with this system are just absolutely phenomenal. So looking at blue and orange, this is our V1 Cas12a protein delivered with a variety of different guides as RNP. And then taking the same guides and switching to our V3 product, you can see it's just an absolutely night and day difference. So we've really made significant improvements with this system. And then we've got the same data plotted in a box and whiskers plot on the right-hand side here. Again, blue and orange are different replicates of our V1 offering, and in gray and yellow are our V3 system. So again, this is our V3 Cas12a, or formerly known as CPF1 protein, and this V3 product will be available at the end of the month. So very, 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 very shortly. So what should we take home from this talk? Well, RNP delivery of Cas9 and Cas12a systems, I think, are the way to go. It simplifies everything. You eliminate the complexity from immune system stimulation and toxicity, and you can dramatically reduce off-target editing without using high-fidelity proteins. For Cas9, I think you can comfortably use the HiFi protein to ensure that you're going to get good on-target editing and exceptional reduction in off-target editing. And we've improved that even further by putting it in the context of our V3 proteins, where you're getting exceptional performance and you're dramatically increasing editing efficiency in live cells. And for the context of HiFi, you're not doing that at the expense of reducing off-target editing. And again, all of these things will be available on our CRISPR website at the end of the month. So with that, I'd like to thank everybody for attending, and I'd be happy to take any questions.
Thank you, Chris, for that very informative presentation. We will take the remainder of the hour to answer any questions that have come in. The first question from Kelly is, do shortened guide RNAs work as well with the IDT's HiFi Cas9?
Yeah, so we don't recommend using shortened guides with wild type or HiFi because it seems to be very context and site dependent, whether they work or whether they don't work. And I think the HiFi is slightly more susceptible than the wild type, but in general, I wouldn't recommend using shortened guides with either.
Thank you. Next from Stephen. How does HiFi Cas9 compare to the HiFi Cas9 with on and off target activities when delivered as RNPs?
Okay, so yeah, we've actually looked at a lot of these. We looked at HiFi, we've looked at the newer Evo Cas9, and we've also looked at X-Cas9. And what we found is that it's sort of the same story where they're very, very, very good at reducing off-target effects, but when we test them as RNP, the on-target editing really suffers. And I would say that HiFi, Evo, and X-Cas9 all produce lower on-target editing efficiencies than the two that I showed today, the ESP-Cas9 and the SP-Cas9HF1.
Okay, next, William would like to know how valuable are programs including IDTs that predict the guide RNA efficiency?
I think they're extremely valuable. I just think it's such a difficult thing to do that, you know, I think eventually somebody's going to get it right and it's going to really improve things. But I just don't know how much work it's going to take to get there. I really think it's turned out to be something that's really, really challenging to do. So to answer your question, I think it's invaluable.
Right. Next, Felix would like to know why you focused on using GuideSeq rather than CircleSeq in the presentation.
I think that, you know, there are many ways to do this, and, you know, each has its advantages and disadvantages, and GuideSeq is one of the first to come out, and we latched onto it because it allowed us to analyze global off-target editing in living cells. And for that reason, we chose to, you know, because there's a lot of development that goes to even reproducing things that happen in the literature. So we put our effort into GuideSeq, and we found what worked for us internally, and we just kind of stuck with it and incorporated it into what we thought is the best overall approach to look at off-target editing. So, you know, I think any of those different sites, CircleSeq, Genome, what have you, I think they're all, you know, they could all be used and produce similar results.
Great, thank you. We've gotten a couple variants of this question. What changes were made between V1 and V3? Were they adding more mutations or optimizing expression or purification protocols or NLS and etc.?
Okay, so what I can say about those is we did not make any additional substitutions to the native ORF. These are changes to the region's flanking tags, linkers, and nuclear localization signals.
Great, thank you, Chris. Elena would like to know if we have noticed any change in Cas12a V3's off-target profile.
That is a really, really great question, and we're just going to have to look at it. It's something we haven't looked at very closely yet, but no, it's a great question. I think we were so blown away by what we made that we hadn't sort of gotten to that yet, but it's a great question and something we will address.
Yes, absolutely. We have a question asking if we have tested the immune cell activation with RNP. You show at the beginning the activation with IVTs, but we haven't seen any increased activity when using RNP at all with immune cell activation, I think is what this question is getting at.
Okay.
Next, Theodore would like to know if there's any indication if the cRNA length can alter the cut site position.
Well, that's an interesting question. It's just not something we've specifically looked at, but I know that we've shortened the cRNA and we're able to look at repair profiles and all that sort of stuff for any of these experiments, and you would expect that the indel profile would probably change if you're going to change the cut site position, and we've seen no evidence of that. All we really see is a change in the frequency of indel formation. We haven't seen any of the changes we've made. We haven't seen any difference in the cut products or the frequency and repair outcomes.
Great, thank you. Liam would like to know if the RNP system is effective for modifying bacterial genomes.
Yeah, so the answer to that question is I don't know. I do know that bacteria have extremely powerful 3' to 5' exonucleases, and I also know that it's not a super common thing to try and deliver protein directly into bacteria via electroporation or chemical methods. I do know there are reports of people getting fluorescent proteins in. I've never seen anybody get an RNP in. That's something we've talked about trying to get to work. But no, I just don't know the answer to it. I don't know if anybody's done it yet.
Right. We have a question if we are doing any work to reduce the size of the Cas9 protein.
The answer to that question is we're not doing anything to reduce the size of SP-Cas9. I don't know that it's extremely modular, and I don't know what we could give up and mix and match without completely disrupting its function or making unintended changes. But we're always looking into new systems, and I think if somebody is to find a similar Cas9 from perhaps a different species that is smaller and gives the same quality of editing efficiencies, that would probably be ideal. But no, it's not something we're actively working on.
Thanks, Chris. We've got a few more questions that are coming in. Natalie would like to know if it's okay to increase the Alt-R Cas9 concentration to increase efficiency. So increasing the RNP concentration to drive up your activity.
Yeah, I think absolutely. It just depends on, every system is going to have limits in terms of what is and what is not toxic. But I think it's definitely a reasonable thing. In fact, every time in the laboratory we look at a new guide, typically we'll do a dose response going from high to low, looking at both on and off target. So I think it's a totally reasonable thing to add more and try and stress your system and find out, where your particular guides and RNP complexes are toxic or not.
Great. Axel would like to know if you could provide any advice on how to optimize the Cas9 complex for HDR rather than non-homologous end joining.
Yeah, so Ashley, do you know if we have the app note online yet for HDR?
We do. So Chris is referencing an app note that you can access on our support link on our website that's going to provide some basic information for delivering a short single-stranded oligo, which is an ultramer for HDR that shows high activity with RNP.
Yeah, so there are a lot of questions. HDR is obviously more complicated. Do you use enhancer? Do you not use an enhancer? What concentrations do you use? And I think the best thing I can do is direct you to that app note on our website.
Yes, and we are actively working on ways to improve HDR as well, and we'll have more information on that as it comes out.
Yep, absolutely.
Dale would like to know if IDT's HiFi Cas9 can tolerate a heterologous G at the beginning of the guide RNA.
Heterologous G at the beginning of the guide RNA. So I guess if you're using an in vitro transcribed sgRNA and having a non-templated G.
Correct.
I mean, I think it's going to be, I wouldn't recommend doing it. I think it's better to use the chemically synthesized system where you know what you're putting into cells. But I mean, you could empirically test it and try. I just think it's going to be kind of a mixed bag of what sites work and what don't. And the same is going to be true for wild type in some cases. So I think I would recommend using a chemically synthesized two-part system or single guide RNA where you're not introducing non-templated nucleotides.
Great, thank you. Another question that has come in is if the V3 proteins still require using the Alt-R electroporation enhancers.
Yeah, that's a really great question. I think there are still a handful of sites, especially with Cas12a, where the overall editing is low and you still see a pretty significant jump with the enhancer. So for Cas12a specifically, I think the answer to that is yes. It's still something that should be put into practice. It's certainly not going to hurt. For Cas9, we're saying it's not a requirement, but we still recommend using it. And I think on the outside chance that you've picked a guide that is going to give poor editing efficiencies, regardless of what protein you're using, it's still not a bad idea to use the enhancer. It's not super expensive, and I don't think it's going to hurt at all. So I think the answer to that is yes, we're still recommending using them.
Yes, and all of the other Alt-R products will work seamlessly with the V3 proteins.
Yep.
There was a question if we will still offer the V1 proteins.
So they're going to not be available on the website, but we understand that there are certain people that are maybe in the middle of an experiment where they need to use the exact same protein, and if that's the case, you can simply call our support line, and we will have limited quantities available for those customers for, again, a limited period of time.
Great. Christina has a question. She's asking if you can explain again what the difference between Cas9 and Cas12a is. Maybe there's some confusion with the new nomenclature switch of Cas12a to CPF or CPF1 to Cas12a, but briefly describe that.
Yeah. I kind of figured that out giving this webinar, is that maybe it should have stayed CPF1. But yeah, so they're very, very different enzymes. Cas9 from a different bacterial species, from Strep pyogenes, recognizes an NGG-based PAM that's located at the 3' end. It's natively targeted by 2 different RNAs, whereas CPF1 or Cas12a, we use the version from Acetomenacoccus. It targets an entirely different triple T VPAM that's located at the 5' end of the targeted sequence instead of the 3'. Even the nature of the cuts is different between the two enzymes. Cas9 cuts blunt, whereas Cas12a or CPF1 leaves staggered in. So they're 2 very different RNA-guided DNA endonucleases.
All right. We're going to do one more question from Dale, who would like to know what the shelf life of the HiFi Cas9 protein is.
Oh, great question. So we've tested this and we haven't seen any difference between our HiFi and our wild type. And I think, we've tested this in so many different ways, looking at 4 degrees stored in liquid, minus 20 in liquid phase. We've kept it at minus 80, and I think it's still shows 100% function out to a year. I don't recall off the top of my head what the official recommendation is, but we don't find that HiFi is any less stable than wild type. Great question.
Okay, that is all the time we have for questions. I want to thank all of you for attending today's presentation. I also would like to thank Chris for his informative presentation, as well as Ashley for conducting the question and answer session. This is one of a series of webinars we will be presenting on CRISPR, as well as other topics. We will email you about these future webinars as they are scheduled. Also, as a reminder, a recording of this webinar will be posted shortly on our website and at youtube.com/IDT-DNA bio. There you will find several other educational webinars on such topics as genome editing, genotyping, next-generation sequencing, qPCR, and general molecular biology. Also, the presentation slides for today's webinar have been published on our SlideShare page at slideshare.net/IDT-DNA. Thank you again for attending, and we wish you the best of success in your research.